All right, so thanks everyone for coming. So I'm going to be talking about how to read minds using Fmri. It's sort of an overstatement, maybe, to say reading minds, and it's not like you can put someone in a scanner and just have them think something and then figure out what they're thinking. But to some degree, reading minds is possible, and perhaps that'll become more evident after we go through the encoding model. But since different people have different experience with Fmri, first I'm just going to go through some basic overview of different analyses. So back in the 90s, early 2000s, people were using univariate approaches to Fmri. So people would be in the scanner, and you would be able to detect changes in blood flow to regions of the brain that were more active. So people were able to measure the blood oxygenated level-dependent signal. And so if you had, say, a region of the brain that represents places, so the parahippocampal place area is known to represent places, and you presented someone with a place, a scene, a building, then the baseline firing rate in that region would increase, so the bold signal would increase, and we would be able to detect that using Fmri. Likewise, in a region that doesn't represent places, like the fusiform face area, which represents faces, there would be no change in the baseline bold signal. If you presented someone with a face, though, and you were to measure the same regions, then you would see an increase in bold signal for FFA and not PPA. Then multivariate approaches came about. So you were then able to not only just look at a region of interest and see whether the bold signal has increased or decreased, you can answer questions about the specific pattern of brain activity within that region. So if you were to present two different faces, for instance, the bold signal would increase in both cases in the same region, and so a univariate approach wouldn't be able to get access to that. So within a region of the brain, you have a lot of voxels being the unit that we are able to record from. So a voxel contains thousands of neurons, and within an ROI, you can have 100 voxels. It depends what the ROI is. But by looking at the pattern of activity within that region of interest, you can see more finely what voxels are having a change in activity. And it's this pattern of activation that can get us answers to questions that are more subtle than univariate approaches can handle. So for instance, let's say that somebody had seen 50 faces of Ben and 50 faces of Molly. We could build a classifier in such a way that a certain pattern of activation would be more similar to Ben or more similar to Molly. So if we present a new trial, and let's say that it's Ben, and we look at that pattern of activation, we can say, okay, that is more similar to the past 50 Ben trials than the past 50 Molly trials, and thus we can classify that stimulus as being Ben's face. What happens, however, if we have a novel stimulus? So for instance, let's say that we don't have 50 trials of someone's face to have constructed this classifier out of. Then things get a little bit more tricky, because if we only have one trial worth of this person's face, then we can't say, is it more similar to something that we haven't classified yet? And that's where decoding sort of falls apart. So in the email that I sent out, I gave a link to a study from Nishimoto and colleagues where they were able to reconstruct the movies that people were watching in the scanner. And I mean, the reconstructions are pretty grainy and low quality, but it's still amazing that they're able to even get something that somewhat resembles the original movie clip that the subjects were watching. And this is still decoding. So, in other words, looking at pattern of activation and being able to say is it similar or different to something else. More specifically, these subjects watch hours of movie clips. And at the top left, we have the presented movie clip, and then the 3 rows represent 3 different subjects. And based on the pattern of activity for each of the subjects, the authors could look back and say, what is this pattern most similar to, given all of the other clips that they've watched? And by averaging together all of the most similar clips, they were able to get a reconstruction of what the original movie clip was. So in this case, they took the 100 most similar clips and they just fused them all together, they overlapped them all together, averaged them, and then they got the result at the bottom right. If they hadn't averaged it together, they'd just get the most similar clip at the top left. So this has all still been decoding, so what even is encoding? Well, encoding allows us to ask, how does the brain represent stimuli, as opposed to what stimulus was someone looking at given their brain activity. So that is, just to repeat myself, encoding uses stimuli to predict brain activity. Decoding uses brain activity to predict the stimulus. So they're basically opposites of each other. And a recent article from Popov and colleagues gave a nice overview of what encoding means. And if you look through the literature and try to look at different articles that have used encoding models, it can be somewhat difficult to navigate. So if you see inverted encoding models, forward models, voxel-wise modeling, stimulus-based encoding and decoding, voxel-based encoding and decoding models, these all represent the same underlying technique. There are some differences. So inverted encoding models, they always invert a matrix, and so it's the computation involved that lends to the name. But generally speaking, these are all the same. They're all using encoding models, so I'm just going to be using the term encoding models. So let's get into some actual fMRI data to better understand things. And by the way, as always, feel free to type or chime in voice chat, whatever works, if you have questions or comments or anything. So I'm going to be using data from, or using a tutorial from Tommy Sprague. He was a former grad student in John Sorensis's lab, currently a postdoc in Clay Curtis's lab at NYU. And the tutorial makes use of data that was obtained from Mary Smith, who was also a former graduate student in John Sorensis's lab. I also want to thank Julie Gollum, who is my advisor. She added comments and additional code to the tutorial that was helpful. And I've also asked many questions to Emma Wu Dowd, who is a postdoc in Julie Gollum's lab and who has actually used encoding models in her work. I'm just trying to start an fMRI project that uses encoding. I don't have hands-on experience with the technique. So she has been generous enough to answer questions that I've had. Just a second. All right. So in the tutorial, the data, they make use of a very simple technique, and that is an attention task. They present a gabor, which is an oriented grading of black and white lines. They present 2 of these gabors, one on either side of a fixation dot, and on every trial, the subject in the scanner is supposed to be attending to either the left or the right side of the screen. So they're attending to one of the two presented gabors. The empirical question was whether the reconstructions would be better given the attended gabor or the unattended gabor. Could you even reconstruct the unattended gabor? So in the files that you can get from the GitHub site there are 3 different files. One is the bold signal for every voxel in area V1, early visual cortex. And there is the bold signal for every trial. There's also a file that lists the orientation of every gabor for each trial, and also what side of the screen was being attended for every trial. So I'm just showing the different data files here visually. So for instance, here are 5 trials with the bold signal for all of the voxels. Here are the respective orientations of each gabor for every trial. This is only looking at the left hemisphere. So because they wanted to separately analyze the left from the right hemisphere, that's why you don't see 2 gabors on every trial. This is only the gabors presented on the left side of the screen. And the blue and the red dots correspond to what side was being attended. Red means that they were attending to the left. Blue means that they were attending to the right. So the first step in building an encoding model is to specify your hypothesized underlying representational content of the region of interest. So in this case, they created a basis set consisting of different orientations. So I also need to point out that all of the gabors could only be presented in one of 9 orientations: 20, 40, 60, 80, 100, 120, 140, 160, 180. And so each of these corresponding orientations was modeled as a separate gabor, as a separate normal distribution, a Gaussian. More specifically, it was a sinusoid raised to a high power, but generally speaking, the exact shape of these distributions doesn't matter that much. So each of the orientations was separately modeled, and they have their basis set represented as a matrix. So you can see in this matrix that there are 180 rows. Each one of these rows corresponds to an orientation, so a degree of orientation. And so for instance, I said that 20 degrees was one of the possible orientations for the gabor. If you look at row 20, you can see that the first channel of the 9 columns, each column is a different channel or basis sinusoid wave that I showed. So if we imagine that the 20 degree mark is represented as the first channel, then the highest response or the amplitude of this filter would be a 1, so it's unchanged. But the farther you get away from this channel, the less important the signal is, basically. So it's a decrement in the amplitude of the filter that's going through. And that'll hopefully make more sense later on as I show you what we're actually using the basis set for. So, just as an example, let's say that one of the trials, trial 5, the left gabor was oriented at 120 degrees. So that would be represented by the light blue Gaussian. And on the right, you can see that that's just the spike where the orientation is. And so ideally, it would be represented fully as the light blue Gaussian and represented less fully by the green and by the red, and even less fully by the purple and the dark blue. So if we were to model that as a predicted channel response, it's unlikely that we're going to get just, a pure spike in amplitude at the exact channel that we predict. So because it's modeled as a Gaussian, it gives us some leeway in how the signal is going to be interpreted. So in other words, greatest importance to the top of the light blue Gaussian, and then as you go farther away, this filter makes it so that it's less important, which is shown. The channels are completely theoretical, yes. These are, basically we're trying to construct the underlying representational content of the region of interest. Right now, this is not looking at the neural response or the fMRI data at all. So it's basically trying to create an idealized view of how that area of the brain is representing orientation. So assuming that V1 corresponds to orientation, we can predict that it can be ideally represented by these different channels. And if we linearly construct them in a way so that if it's represented by like 120 degrees of orientation, it's going to be most fully represented by the light blue distribution and not as fully represented by the other ones. So on the right here, we have a design matrix, which is just another way to visualize the second figure. So in this case, the white represents the peak of the filter, and then it levels off as you go away from the center of this idealized orientation. And we can do that for every single trial, which looks like this, and for both the left and the right hemispheres. Again, I'm sorry that this is very hypothetical and theoretical right now, but this is how the encoder gets built. Basically, what happens now that we've constructed this hypothesized underlying representational content of the ROI is that we train the encoder on fMRI data. So for every run that these experimenters collected, they're going to split the data in half. Half of the trials will be training and half of them will be test. So as part of the matrices for the training and the test, there are 252 trials in the training set and 105 voxels. So each voxel is associated, or there's data for every voxel for every trial. So 252 trials for 105 voxels. And then we also have idealized channel responses that we were talking about before. So for instance, we have 288 trials for the 9 respective idealized channel profiles. You'll notice that there are 252 trials on the left, 288 trials on the right, and I will get into why there's a difference in the number of trials. But basically what we're going to do is we'll take the bold signal for every voxel for every trial, and we're going to take the idealized channel responses and divide by that bold signal. And that's going to create a weighted matrix. So the theoretical reason behind this is basically you have bold signal for every voxels for every trial, but we also know that the bold signal is corresponding, we think, to a certain orientation, which corresponds to a certain Gaussian in our hypothesized distribution. So we're going to more heavily weight the influence of the bold signal according to the orientation that it actually was. And that weighting corresponds to this matrix on the right, whereby 1 means that there's no change in the bold signal, so it's fully represented, but we're not going to pay as much attention, we're not going to weigh as much to the orientations that are farther and farther away from what the actual orientation of that gabor was. So this is the actual math. It's literally just dividing the right by the left. So if we do that, we can look at what we predict would be the response for a certain trial. So on the top, we see the sample channel weights for 3 of the voxels. I could show all of the voxels, but it wouldn't fit in the plot very nicely. And if we average together across all of the voxels, we get a predicted channel response for that trial. And so in this case, the predicted channel response is that the gabor is 40 degrees in orientation. And we know that because that's how we created the model. It should be where the orientation of that trial was. So in this trial, the gabor that was attended on the left side was oriented 40 degrees. All right, so this yields some cool things. We've basically been able to predict the weights for every voxel for every orientation. And at this point, we would like to compare the channel weights to the actual beta weights, the bold signal, for each voxel for each trial from the test data set. And this should ideally predict the orientation for each test trial. And this is how we're going to test the encoder and see if it's actually working, by using the model that we've now just created on data that were not used in the training of the model. And the method for testing is using a leave one, run out cross-validation. So basically, we're going to be training on all of the runs aside from one, and we're going to be testing on the one run that was left out, and we're going to do that iteratively for each of the runs. Do the voxels in the data set include only the ones in the renotopic location of the gabors? No, this is purely area V1, left or right hemisphere. There's no additional mapping of the spatial locations of the gabors. So we're using leave one, run out cross-validation to make sure that the model is correctly estimated. And then we will test the encoder on data that were not used to train the encoder. And we do this by inverting the encoding model. So how do we invert the encoder? Basically, we have these different matrices. We have the weighted matrix that I showed before. This is 105 voxel by 9 channel matrix. And by inverting that, we basically get the reciprocal of the learned weights. So, basically, like, it's not exactly a reciprocal, but you can think of it that way. Like, if the weighted matrix was equal to, say, the number 8, then we would get 1/8 at the end. So it's literally just trying to invert it to get the reciprocal, theoretically. And we are then going to multiply that by the following matrix that we compute using the test data. So we take the weighted matrix that we've created before, and we multiply that by the test data set. So the weighted matrix, 9 channels by 105 voxels, we multiply that by the test matrix, which is 105 voxels by 36 trials. If we multiply the two matrices together, we get a new matrix, which I'm calling B. And this is a 9 by 36 matrix, so 9 channels and 36 trials. And this represents the test data set, bold signal, that has now been weighted properly based on what we've derived from the original training encoder model. And then we multiply these two matrices together, and that yields the reconstructed channel responses. So for every trial, we get 9 channels. Or we have the 9 channel data for all of the 288 trials. I forgot to mention the leave one run out cross validation. That's the reason why we had 255 trials instead of the full 288 2 slides ago. So when training the model, we actually don't use all of it. We train it on the one run left out. All right, so after we've inverted the encoding model, we have the ability to decode or reconstruct trials, data that were not used to build the encoder. And so I showed you the top and bottom figures from before, but the middle figure is the reconstructed channel response for one sample trial in which we've averaged across all of the voxels, and this is the estimate that's been yielded based on the encoding model. And you can see that it's somewhat related to it. It's not perfect, and other trials will be more or less better in reconstruction. But what we then plan to do is to collapse across all of the different orientations that were possible in the test data set. So here we're circularly shifting all of the orientations so that the center of the channel is at the 100-degree mark. So this way, for trials in which the gabor was 20 degrees, we've shifted them plus 80 degrees. And this way we can average all of the reconstructions at the same point to see how well the reconstructions are, basically. So we can see that the reconstructions are doing quite well. It's centered at the correct location, and therefore the encoding model is doing a good job because it's able to decode stimuli according to the original orientation. So this is great, and I hope it wasn't too complicated, but we haven't really been able to decode novel stimuli in this example, right? So all of the gabors were at 20-degree intervals. And what I said before was that the encoding model is useful because it's able to decode novel stimuli, right? So let's go to another example. In this case, they presented oriented gratings for 1 second, and then the subject in the scanner had to, and again, stop me at any point if things are getting confusing, that the subject had to keep in memory the orientation of this grating over a delay of 12 seconds. And then they had to, well, they were tested on what the orientation was. So this was, they're represented with a grating, and they can rotate the grating to match what the original stimulus was. And what the authors of this paper wanted to do was to decode what the orientation of the gabor was during the delay. And unlike the tutorial that we were going through, these gabors could occur in any orientation. So 1 degree, 2 degrees, not just the 20-degree intervals from before. So one way to think about encoding and decoding is to separate things into 3 different components. Hold on. So presumably some voxels in the data set would respond more to the gabor. Yes, that's totally correct. But yeah. So the 3 components that could be thought about in terms of encoding and decoding are an input space, a feature space, and an activity space. So the input space is literally just what the stimulus is, so the pixel information, for instance. And the activity space is just the voxel activity, the bold signal. And the intermediate space is the hypothesized representation. So in this example, it would be orientation again, represented by the same basis functions. And in an encoding model, looking just at the top row, it follows this left to right pattern. So you have the pixel information, you have the stimulus. This can be represented in an idealized feature space of 6 different orientations in this example. And from a linear summation of those weights, we can construct any stimulus. So, for instance, in this example, let's imagine that it's negative 10 degrees, I guess. The different weights for the different channels This is all arbitrary. I made these units up, but the yellow distribution, it has contributed the most, and the far left and right distributions contributing the least. If we simply construct a Gaussian distribution that most effectively is represented by these 6 weights, we would then center that distribution at roughly the -10 degree mark, like it's showing in this example. And then we use the same approach as in the tutorial, construct an encoding model, use some form of training. You can use one run left out, cross-validation, or other measures as well. And this yields a predicted bold signal. So it's going from left to right. The encoding model is constructed from the feature space and then used to predict what the bold signal should be. In contrast, decoding goes in the opposite direction. So you have bold signal, you have activation, and then using that bold signal, you can go backwards and invert the encoding model to figure out what the orientation is predicted to be. And if you are able to predict that orientation, you basically know what the stimulus is. Now, without an encoding model, you're not able to do decoding based on the encoding model, of course. But basically, you won't be able to have these different weights that correspond to it. And so decoding is simply referring to going backwards from activity space into feature space, in which case you can figure out what the stimulus was. Encoding models are good because you're able to decode according to this underlying representational content. Without an encoding model, you can still decode. It would just be based on, say, a linear classifier or something else. But basically, encoding models always entail decoding because that's how you can verify that your encoder is working. It's just this kind of confusing terminology where encoding-based explanations necessarily always deal with decoding at the end, but papers that use MVPA and univariate analyses are decoding-based without the encoding model that's constructed in the middle. So now I wanted to move on to the original example of the movie clips from the Nishimoto and colleagues paper. So this is a bit more complicated, but it follows the same sort of logic. So they have an input space, which is a movie clip represented in red, green, blue pixels. And then they use a nonlinear transformation to derive this somewhat complicated feature space. They use spatiotemporal gabors. So I'm not going to get into the exact nonlinear transformation that they use, but needless to say, it involves trying to calculate gabors at different locations on the screen. They actually use 6,555 gabor filters, but I represented this using far less gabor filters. And the gabors were also able to vary in position, orientation, direction, spatial and temporal frequency. And then after creating this insane feature space of spatiotemporal gabors, they then linearly transform that in the same way as was used in the tutorial into an activity space, the predicted bold signal. And when they wanted to reconstruct the movie clips, this is again going into what decoding is. So they have the bold signal, they invert their encoding model, although not in the same computational transformation that was used in the other papers, but they're using the encoding model in reverse. And that predicts what the different filters are in the feature space. And based on that, they average together the most similar movie clips in order to get a rough approximation of in this case, the Steve Martin clip but whatever the stimulus that they were looking at was. I also wanted to point out back to the Popov and colleagues paper that was recently put out, is that there are some pitfalls to encoding models, and people have made some really far claims about encoding models. So if the brain uses this representational scheme, right? Let's say that V1 is represented by orientation, right? So an encoding model that implements those features should and will be able to predict neural activation well, which makes sense because we're correctly representing that region of interest with a feature space that adequately represents what that region of interest is doing. However, it's important to note that successful prediction using an encoding model doesn't guarantee that the brain is actually implementing that representational scheme. So like I said in the beginning, when we're constructing this basis set of the different channels and everything, this is not using fMRI data. It's completely hypothesized and idealized, and just what we think is how that region of interest is computing to certain stimuli, right? But just because we're able to correctly predict something doesn't mean that that area of the brain is actually implementing our idealized representational scheme, right? So there was a paper that came out that these authors pointed to. The authors were trying to understand how concepts are represented in higher order semantic brain regions. And so they constructed this encoding model that was based on subjects' ratings for 5 sensory motor features for different words. So subjects would see these different words, and then they would rate them according to things like color, motion, sound, shape, and action. So their encoding model was based on these different features, right? And so they were actually able to predict neural activation patterns for novel words that subjects saw in the scanner. But does that mean then that the region of interest that they were looking at, this higher order semantic brain region, is actually representing these words according to color, motion, sound, shape, and action? The authors claimed that the region represents concepts as this multimodal combination of sensory and motor representations, but that's not necessarily true. Just because there was accurate prediction doesn't necessarily mean that that's how the region of the brain is representing the stimuli. So what the authors suggest is that you can make these claims about underlying representational mechanisms, but you need to have more evidence in order to do so. So for instance, in the gabor example, we saw a graded pattern in the reconstructions that peaked at the correct basis channel, right? That could be evidence for the representation actually being based on observation. In this word higher order concept study, they didn't show like a graded degradation from color to motion or something. So they weren't able to then show that. And you can also look at the reconstructions and see if there's systematic errors going on when the reconstructions are not correct. And if there are systematic errors, then that can tell you that something is wrong. So, for instance, in the grading example, they saw graded patterns in reconstructions, and they were then able to conclude that that they were able to support the sensory recruitment hypothesis that the area of the brain that represented the stimulus during a delay was recruiting the area of the brain that was used to represent that stimulus. And so it was valid for them to make that claim. But it was only because they had further evidence than simply they were able to predict the stimulus more often than chance. And that's all I have. So thank you for tuning in. And let me know if you have questions or comments. I know this was a bit complicated, and even I'm not an expert in this, so I might have gotten a couple things wrong, but I'm very interested in the technique. I'm trying to learn it to the best that I can, and I think it has a lot of cool further studies that can be done from them. Yeah, need to learn a lot more. Me included in that, I'm not sure. Any recommendations for what? Like how to learn the technique? Yeah, so go to the GitHub site and play around with their code. I'd also recommend reading the Popov article as well as Nishimoto and colleagues, not 2011, not the movie one, but they have a general overview. Oh, it's not Nishimoto, it's Nasalaris, K, Nishimoto, and Gallant, titled Encoding and Decoding in Fmri, which gets more at the differences between input space, feature space, activity space. I think that's a really good read. Wow, Molly. Yeah, I mean, I'm interested to know what future discussions should be about. Oh, I also wanted to point out that next month we have Ayala Alan presenting her work that she did at Tel Aviv University. And the month after that, we'll have Stephen Adamo, who's a postdoc at the George Washington University, giving an overview on his recent stuff that has to do with visual search. So stay tuned for that, and join the mailing list if you haven't already. You can find that at the website.